Thanks, Irene. 50 town halls, that is a milestone I didn't know I was trying to hit. Seriously though, FDA has found these to be very helpful as a way for us to engage with stakeholders, and hopefully you all have also found this to be very helpful. So thank you again for joining us this week and for the past 50 weeks for many of you. So I have a couple of updates today, and then we'll go through some of the questions that were sent ahead of time and then open the line up for live Q&A. So last week, right after last week's town hall, we issued EUAs for 5 serial screening antigen tests. I guess it was really 3 tests, one of them in a few different configurations, but 5 EUAs. We then issued another serial screening antigen EUA, I believe the day after. And since then, we have also issued 1 serial screening molecular test EUA. So those are all based on the serial screening supplemental template that we issued a couple weeks ago, and we talked about on this call. And we're pretty excited that developers have been picking that up, and we've been able to get several authorizations out under that approach. And we know that there are other developers that are pursuing that, and we look forward to keeping the momentum going there. We think this will be very helpful for the serial screening programs that we know are being stood up around the country. We also, since the last town hall, issued an EUA for the first antibody test that can be used with home collected specimens. So that test can be used with finger stick dried blood spot samples and sent to a lab for processing. And then just this week, our COVID testing basics webpage was updated. So that's available on the FDA website and provides a lot of good background information on coronavirus testing and the different types of tests, when to get a test, things like that. Probably all things that the developers who are our typical audience on this call already know, but it's a good page to share with folks who may be interested in that level of information. So with those updates, I will move on to the questions that we got by email. So starting out, we got a couple of questions about validation studies for multianalyte molecular tests, looking at coronavirus as well as flu, RSV, et cetera. And the concern with acquiring archived influenza specimens, particularly, is one of the questions that we got, and asking whether FDA would consider the use of contrived influenza specimens for an EUA submission, with the expectation that a prospective clinical study would be conducted post-authorization. We do know that there's been some concerns with the ability to obtain archived specimens, and if you are running into that roadblock, we encourage you to reach out to us to discuss your specific situation. But for the most part, we do continue to recommend the use of archived positive and negative clinical specimens rather than contrived. We know that they may be difficult to obtain, but that prospective testing is going to be with actual clinical specimens is going to be generally what we expect for those types of studies. We have another similar question about studies for the same type of test, and also getting into studies that would support a potential 510K. We do generally recommend that you evaluate clinical performance of those multi-analyte tests with a prospective clinical study. and we do, for EUA, we have indicated that a single site using archived specimens would be sufficient, and you may want to consider confirming the sample positivity due to possible degradation over time. There was also a question in that same one about specimen types and whether it was okay to use a single swab to obtain an OP sample and then also an NT sample, so that the specimens tested would be OP-NT, and asking whether that would be acceptable for a 510K submission. So we do accept specimens that are a mixture of, or samples that are a mixture of specimens. But if you're seeking an upper respiratory claim for individual specimen types, we do recommend that that each specimen is individually collected, and we generally consider NP swabs, nasopharyngeal swabs, to be the most challenging upper respiratory matrix. Chris, do you have anything to add on those questions about multi-analyte tests?
Sure. I would say that there's a long documented regulatory history for flu and RSV and those types of panels, and there's numerous decision summaries that are publicly available for analytes that are not in the SARS-CoV-2. And there's a lot of information there, and a lot of questions can be answered by looking at previous decision summaries.
Thanks, Chris. That's a really good point, that all of that information is available on our website and is very helpful for test developers. Okay, so moving along, we have a couple of questions about the Biofire de novo. We do have a question about whether the decision summary for Biofire will be posted, and it will be. We unfortunately are not able to comment on the timeline, though we are working on getting that out. I'm sure everyone is aware that decision summaries for de novos sometimes take some time to get posted, but we are working to expedite that one. We also, I think we discussed this a little bit last week, but there's a question about the Biofire being challenging to have access to as a predicate, since it's the only de novo currently and the only predicate for a future 510K for SARS-CoV-2 tests. And we did talk last week that you can use an EUA authorized test as a comparator for your clinical studies, even though Biofire would be your predicate for the submission itself. There was the question also was asking about whether there are other options, such as testing to the reference panel, to support a 510K. And while we do consider another EUA authorized comparator to be acceptable, we do really want to see that the prospective clinical study in your 510K submission. You can also include reference panel results and the clinical data from your authorized EUA if you have one, and leverage those, but they would not be generally they would not be considered to be sufficient on their own to support a 510K clearance. Chris, anything to add on that one?
Sure. I think in this time between while we're waiting for that decision summary to be posted, the granting letter is posted, and it does have the special controls noted for these types of tests, and you can kind of take a look at those special controls and design your studies to meet those requirements as well.
Great, thanks. So moving on, we have some questions about asymptomatic screening and particularly related to the supplemental template that we put out and asking about the post-authorization validation study. Generally, we would want to see your proposal for a serial testing plan and the post-authorization study in your EUA request. And we would discuss that with you during the review, particularly the statistical plan, which is what this question is focused on. We would want to go through that with you in detail, and so we can't really get into too many details on the statistical plan on this call because it will be specific to each individual test and each approach that you're looking to take as a test developer to validate your test. And then along the same lines with serial screening using that supplemental template, we have some questions about the additional data that we would expect to see. And so, the way that supplemental template is laid out is that it is there as an approach so that if you have symptomatic validation data and you've been authorized for testing symptomatic individuals, that template allows you to expand or to request expanding your claims to asymptomatic serial screening without doing that additional asymptomatic validation ahead of time. That would be the post-authorization condition. So generally, for most cases, as long as nothing else is changing, so you're not changing your patient population, such as point of care to at home or other changes that might impact the usability, we would not expect to see additional validation, clinical or usability, as long as nothing is changing except for going from symptomatic to serial screening. And we could discuss that on a case-by-case basis if there are things that are changing, whether there is additional data that we would need to see. And then there's another question along the same lines asking about removing the prescription requirement when going from prescription to over-the-counter. And we do have recommendations in the template for OTC use. That's in the non-laboratory use template for molecular and antigen tests. And the supplemental template that we've been talking about also can support this move from prescription to over-the-counter, because one of the requirements for over-the-counter is that there's an asymptomatic claim. We generally would not authorize a test for over-the-counter use if it does not have that broad asymptomatic screening claim, since that's how we would expect it to be used over-the-counter. And so if you are able to use the supplemental template to get that asymptomatic claim, as long as everything else about your test is appropriate for over-the-counter use and has appropriate labeling, usability, user comprehension for a lay user, then we would be able to do that that over-the-counter labeling for you as well in that same EUA request. All right, we have a question about developing a point-of-care molecular diagnostic reader and cartridge system, and what the recommendations are for sensitivity, specificity, and validation testing, also for pooled testing in that situation, for pooled sample collection. So we do include validation recommendations in the molecular diagnostic template for point-of-care testing and for pooling. We have not, as of yet, authorized any tests for pooling at the point of care, and so we would want to have some further discussions about that and how you intend to implement that so that we can make sure that it is a plan that is appropriate for a point of care setting. But generally, we would encourage you to take a look at the template, to look at the the validation recommendations that we have there for the number of specimens and the validation approach. And if you have alternative validation methods that you'd like to discuss, you can reach out through pre-EUA to discuss those. And that question also was asking about whether pooled testing is considered a diagnostic or screening. And I do want to clarify that we have talked a lot about diagnostic testing and screening testing. And we talk about screening when we're talking about testing asymptomatic individuals that have no reason to suspect that they have COVID-19, and doing that broad screening to capture those asymptomatic individuals. But really, a screening test is a subset of a diagnostic test. It is looking to get an individual result and to identify individuals who have SARS-CoV-2 present and have that, have COVID-19. So it is considered to be a diagnostic test from a regulatory perspective. We just generally refer to it as screening to be clear about the indication that we're talking about with the patient population. And Chris, did you want to add anything there about this question about point of care testing or pooled testing?
No, it's just that I think you stated it correctly. I think we have an open approach. We haven't kind of done it yet, but that's something that we could work on in the future. Thanks.
Great, thanks. We have a question about how presumptive positive results should be treated in calculating the NPA and PPA in a clinical study, and whether they should be treated as positive or inconclusive. And generally, we would recommend, and I think this question is talking about the results for the comparator test. So generally, we would recommend that you follow the result interpretation of the test that you're using as the comparator. So the EUA authorization for that comparator test should have a result interpretation section that indicates how those results should be considered. And I think in the Chris, you might want to touch on how we have those listed out in the 2x2 table. I think we do include presumptive positives as positives in the 2x2 table for calculation of PPA. Is that right?
Correct, yes.
Great. Okay, the next question is asking about over-the-counter collection with testing in a lab for PCR testing. So this would be non-prescription, direct-to-consumer home collection, and whether asking about what template to use. So we do have non-prescription, direct-to-consumer home collection tests that we have authorized, and the authorization documents on our website would be a great starting point for looking at how those were laid out and what we looked at. And the question is also asking about whether they can use a hybrid of the over-the-counter home collection kit and molecular PCR templates. And that is absolutely a good way to go. Those templates are all meant to be complementary, since there are situations such as this one, where pieces from each of them would be applicable. And we would suggest that you do pull the parts that are applicable from each of those and include them in your EUA request when you send that in. The question is also asking whether the new supplemental template for serial screening tests can be used, and yes, it also can be used in that situation. And asking about how many participants are required for an over-the-counter usability validation. There is a usability study recommendations in the non-lab template, but that really is focused on tests that are completed that are performed completely outside of a lab, those fully at-home tests, and that recommends, I believe, 100 participants for OTC use. For home collection, you can take a look at the home collection template, which I believe is recommending 30 participants. And if you have further questions about your specific situation, please reach out. And I believe that is all of the questions that we've prepared ahead of time. And so we can open things up to live questions now.
If you would like to ask a question at this time, you may press *1. Again, to ask a question at this time, press *1. Our first question will come from Shannon Clark. Your line is now open.
Good morning. This is Shannon Clark with Userwise Consulting. So I have a very specific question about the human factors validation testing or usability study for OTC. So historically, when we're performing human factors validation testing, we perform self-selection test cases to demonstrate that laypersons understand the outer packaging and aren't purchasing the kit when they shouldn't. Like, do they understand that the product should not be used for children below 2 years of age? So does the agency expect that we run this test case for all users in the study, all 150 participants, that is 50 participants, 50 teams, including users who don't typically need this information? Because we're actually finding in our studies that adolescents who typically are not shopping for 1-year-olds, they generally are pretty unsuccessful with this test case because they don't have a representative level of motivation or attention, similar to what you'd see with parent participants or adults. So in summary, does the FDA expect that we perform self-selection testing in general? First question. And then second, can we proceed with not testing adolescents for test cases that they would very, very rarely perform?
Thanks for that question. So we are interested in user comprehension and making sure that the packaging is understandable for a lay user. I think we are open to approaches that are reasonable in the emergency use setting. And if you have a particular proposal for for what you think is reasonable based on the labeling that you have, we would be open to discussions about that and considering the approaches that you are proposing. I think if you have historical information showing that teenagers typically don't understand this, but also don't need to, that's something that you could include in your submission. I think that would be a very good conversation to have in a pre-EUA specific to your situation, so that we can consider the plan that you have in place.
Excellent. Thanks so much.
Sure.
And our next question will come from Stan Talley. Your line is now open. Stan, please check your line. Your line is open. Okay, our next question will come from Ray Banzulas. Your line is now open.
Hello, thank you for taking the question, and thank you for continuing this series. My question is first some context. My company is developing devices, molecular devices for COVID testing that speak to increasing scale, the capacity of PCR tests in a 24-hour period, and a variety of solutions are in development. The specific question I had related to that is whether FDA would be open to consideration in either a pre-submission or perhaps other kinds of discussion to consider clinical performance data that was generated outside of the U.S. Specifically, we have a device combination that's been in use under the CE-IVD mark in the UK since December, and significant clinical data is being generated and is now sitting with a peer review publication, and we would like to have an opportunity to discuss those results and an opportunity to perhaps discuss an efficient path to authorization for a similar device combination in the United States. Thank you.
Great. Yeah, so we have accepted data from outside of the U.S. For clinical studies. We generally, for point of care, want those studies to be done in the U.S. Because we have found some issues with making sure that that point of care studies done outside the U.S. are truly applicable to a U.S. setting. So in that case, we would want to see them done here. But for central lab tests, we would definitely be open to considering data from outside the U.S. and we would be interested in talking to you about that approach and how how it is applicable to a U.S. population. You may also want to consider including in your submission some information on the prevalence rates in different, where it was studied, as well as the circulating variants, because we may want to see how the different variants are addressed if there are different variants circulating in the place where the study was performed versus here in the U.S.
Sure.
Do you have anything to add there?
No, it's an interesting question, especially the variance aspect of it. As we see more and more genetic variants emerge, this question may become more applicable going forward.
Yes, and so just in terms of logistics, would it be best to put all of that together in a pre-submission format and then request a teleconference discussion on it?
I think that you could include it all in your submission. Chris, would we want to see a pre-sub for that, or just include it in an EUA request and we could discuss any issues as we went through that review?
Yeah, I think at this point we'd prefer to see the EUA request. And if you have existing data, that would be helpful to take a look at as well.
Okay, very good. So straight to the request. Thank you very much.
Sure.
Our next question will come from Susan Sharp. Your line is now open. Susan?
Hi, thanks, Toby. Quick question. If you're looking to add swab pooling to an existing EUA, can this be done with a supplemental submission for the existing EUA? And you can use the molecular pooling template for the swab pooling as well. Is that correct?
That's correct, yes. If there's an existing EUA and you want to add swab pooling, that definitely can be done through a supplemental EUA request using the main molecular template with the pooling information in there. And if you have alternate approaches to validation that you're looking to consider, you can reach out with those as well.
Thank you.
Our next question will come from Annie Wright. Your line is now open.
Hello, thank you for taking my call. We had a question for antigen tests that have been recently approved, and we've heard that they all have mobile apps, and that the mobile app would be required 4 months after EUA approval has been achieved. Is this correct? And also, we wanted to confirm the requirements for the mobile app, which is just to basically ensure that the individual would be able to send the data to their physician. Are there any other requirements for the app?
So the app is something that that we've included as a post-authorization condition for some of for tests that did not have reporting capabilities generally. So this is for the most part related to having the ability to report results to public health authorities. So for laboratories, there is a requirement for labs to report results to public health authorities. That requirement does not exist for home tests, but obviously reporting to public health authorities is very important for monitoring the pandemic. So the mobile apps is one way to enable that reporting from a home test to the public health authorities. And so that's something that we have not required pre-authorization as an authorization requirement for these at-home tests, but we have worked with the developers to put a plan into place to get that reporting capability worked out within an agreed upon time frame after authorization. And I believe that's what you're referring to with those with those mobile apps. Yep.
Yes. And then basically, because our company currently doesn't have any, software engineering or anything like that. So we would be, trying to work with a company. So we're trying to figure out what other requirements would we have to look at for the app. So mainly it's reporting, right? It's basically reporting capability and the data, sending the data to the physician or directly to the state.
So reporting to the public health authorities, I believe, is what we're generally focused on there. Although reporting, the ability for an individual to report to their personal provider is also beneficial. But the public health reporting is really the focus of those post-authorization conditions. And the requirements for those are something that we can work with you on during the review. It's not something that you necessarily have to have fully worked out before you come in. We really want to get tests out, get good tests authorized as quickly as possible. And that's one of the reasons that this is, being added as a post-authorization condition rather than something that we're requiring up front, because we don't want that to hold up the authorization.
Okay. So we wouldn't have to worry about incorporating it into our clinical studies or our usability studies, correct? We would discuss with you during your EUA whether there would be a need for additional usability post-authorization for that. Oh, okay, I see.
Yep.
All right.
But as long as it's not needed to perform the test in your initial authorization, we wouldn't expect to see usability with it at that stage.
Okay. I have one more question. So regarding stability testing, stability studies for home use and also for professional use, is the expectation that the testing should be done in the United States, or can we do it outside of the United States? Because we were having issues with customs and everything if it's manufactured in China.
You said this is for stability studies?
Stability studies. Yeah. No, stability.
Okay. Right. So for stability, that can be done in a lab.
Okay. It can be, and it doesn't matter where the lab is?
Right. That can be outside the U.S. Right. Yep.
Oh, okay.
It's just the clinical and the usability that we really want to see in the U.S. for at-home or point-of-care tests.
All right. Thank you very much.
Sure.
And our next question will come from Richard Montega. Your line is now open.
Yes, we will be running a prospective study for an expanded respiratory panel. And since we already have an EUA authorized coronavirus test with a decent limited detection, we're at 1,800 NDU per ml. Is there any problem with us using our own test as the comparator for the SARS-CoV-2 portion of the study?
Chris, do you want to talk about this one?
Sure. Yes. So for a 510K study, I think the evidence or the performance evaluation is a little bit different than the EUA.
Yeah, we're assuming we'll be running prospective study for 510K.
Sure. For your SARS-CoV-2 analyte and other analytes, is that correct? Or no?
Correct. Yeah.
Right. So you'd have a comparator method that would cover all of the analytes in your study. And for SARS-CoV-2, I think we would likely recommend 2 out of 3 EUA tests. So performing 3 EUA tests on all samples, and 2 out of 3 positive defines a positive. Likely what we would recommend for that.
Okay. Thank you very much.
And that is something that, as you are heading towards a 510K, that we do recommend that you come in with a pre-submission rather, to discuss that study design.
Okay, thank you.
Our next question will come from Susan Sheldon. Your line is now open.
Hi, thank you, Toby and Chris, for doing this. I have one question about the antigen template. It describes the comparator, and when you look at what's posted on the web page, it's difficult to figure out which one meets all those conditions. So I did the best I could and came up with Clarify COVID-19 test kit performed by manufactured by Quadrant. Is that an acceptable comparator for an antigen test?
Yes, I believe the Clarify test kit is an appropriate comparator.
Okay,
and we have,
you.
As we've mentioned, I think on the call before, just for others' awareness, if you do have questions about what is an appropriate comparator, or if a specific test that you're looking to use is an appropriate comparator, you can send that in to the EUA mailbox, and we can get back to you on that as well through that.
Right. Thank you so very much. I appreciate it. Thanks.
Our next question will come from Mark Delvecco. Your line is now open.
Good afternoon, Chris and Toby. This is Mark. I have an administrative question related to the periodic CDRH emails that are put out regarding the status of various diagnostic EUAs. They're usually categorized into 4 separate categories: new, which is obvious what that is, reissued, updated, and revised EUAs. And I'm often asked what the differences are between those, and quite frankly, I don't know what they are. I was hoping you could describe what the differences are between those 3 categorizations.
So I'd have to take a look at the specific emails to know exactly which emails you're referring to. But generally, new is sort of obvious. Reissue is typically when we reissue the complete letter of authorization. That's usually done for things like an update to the intended use. Whether it's going from symptomatic to asymptomatic, or adding pooling, things like that are large changes would be done through a reissuance of the letter. Updates and revisions. I'll see if Chris might have more on the difference between those 2, but there are certain changes that we can grant through what we refer to as a granting letter, and those are included on the EUA tables. If you look at a specific test, there's often a little plus sign next to the row, and if you click that, some of them will have other documents, and those will be a letter granting an EUA amendment in many cases. And a lot of times those are for for smaller changes like an update to the labeling that would not change the intended use in any way. We also sometimes update labeling for things like adding the reference panel results. And so those are some of the changes that would get updated on our website, but don't require a reissuance of the letter of authorization. Chris, I don't know if you have any other thoughts on the difference between updated and revised.
I don't, but I think they generally fall into that category of, the letter is
Sorry about
unexpected.
All right. Thanks. No, I'm just curious because people ask me that all the time, and I don't know the difference. So, but thank you. I appreciate the information.
No problem.
Our next question will come from Sandy Tidy Stevenson. Your line is now open.
Hi, this is Susan Tidy Stevenson. Toby and Chris and all of the folks at FDA, thank you so much for all the hard work that you're doing. We're working with a small company, and I think I know the answers to this question, but they have specifically asked that we ask the agency the following. Whether or not there's an EUA pathway to obtain a quote-unquote PAN EUA authorization for a sample home collection device, where the collection device is demonstrated the required performance with an authorized EUA molecular test, and if they wanted to expand it to be used with other EUA authorized molecular assays, could that be done without additional testing?
So I'm not sure that I fully understand the question. So let me give a little bit of information and then ask for a little clarification.
Okay.
So we have issued a couple of EUAs for collection devices for molecular testing. Those have so far been for saliva. And in that case, those were issued specifically because there was not any legally marketed collection devices for the collection of RNA in saliva. Mm-hmm. And so those were issued under EUA with fairly general indications for molecular testing. And then the assays are, authorized for use with saliva using one of those. So I'm not sure if that's the type of situation that you're referring to, or I'm not quite sure what you're getting at with a pan-SARS-CoV-2 claim for a collection device.
No, I think that you've answered the question.
Okay, great. And I don't know if the device that you're referring to is for saliva or something else. Depending on what it is for and whether there is a need for it from an emergency perspective would be one of the factors that we would look at as to whether or not it would be considered for an EUA. For example, there are a lot of swabs available, and they are 510K exempt generally. So that might not be an area that we would consider for an EUA, depending on the specific situation and the specific device.
No, it would be for something like a dried blood spot sample collection product.
Okay, and I know that for dried blood spots, there are some assay-specific issues. So we would have to look at the specific validation and whether there is a broad claim that could be considered, or whether it would need to be validated specifically with each test.
Okay.
But that's something that we could have further conversations about, specifically about that device.
Okay. Great. Thank you.
Sure.
And our next question will come from, I believe they recorded their name as Bon Say. Their line is now open.
Yeah, hi. Thanks for taking my call. I have a client that's basically developing an at-home COVID-19 neutralizing antibody test. And the question really relies around the neutralizer comparator method that is recommended in the template, the EUA template. Essentially, the client is asking, because of the current methods that are recommended, it's the plaque plaque reduction neutralization test or the focus reduction neutralization testing, and it requires, cells and virus. Can they use for neutralizing comparative method something that's recently been EUA, the GenScript CPAS method?
So unfortunately, I am not an expert on the serology tests and don't have anyone on from the serology team today. So I don't know that I'm going to specifically be able to answer that question, but I think that is something that we would want to discuss in a pre-EUA. But generally, I think that is not a comparator that we would think is appropriate. So we would want to have further discussions. You could send that into the mailbox, and we could get one of the SMEs from the serology team to get back to you on that.
Okay, I already did submit that as a question to the IVD team.
Okay, great. Then I'm sure if they haven't yet, they will get back to you shortly on that one.
Okay, super. Thank you very much.
Sure. Sorry, I can't give you more on that one, but I know the SMEs will be able to.
Okay.
Next question will come from Sarah Berlucky. Your line is now open.
Hi. On a previous call, you had said that unsupervised self-collection of anterior nasal and mid-turbinate swabs in a healthcare facility didn't require an FDA authorization. My question is, does that also apply to saliva collection within a healthcare facility?
So within a healthcare facility, for self-collection unobserved, we do think that you should consider the usability and the appropriateness of array collection without observation, and we would expect that to be part of your validation. So if that's the intended use, that would be how you should validate the test. But we do not consider that to be equivalent to home collection because it does not have the same, some of the same issues in terms of specimen transport and shipping and things like that.
Okay, so that could be applied to any sample type that we were using on our test as long as we appropriately validate.
Yep.
Thank you.
Our next question will come from Shannon Clark. Your line is now open. Shannon.
Hello, this is Shannon Clark again with Userwise Consulting. I just had a question. If we are planning to get OTC designation, but we would like POC designation in the meantime, can we complete the clinical evaluation for OTC and submit that in lieu of point of care clinical studies with healthcare providers? So use lay user data as an input to a point of care only submission as we're working to complete that human factors study for OTC?
So let me start out the question right. So you have a test that is designed for over-the-counter at-home testing, but you want to start with point of care. Is that right?
Mm-hmm. So first of all, just get that point of care authorization, and then 3 months later, once the human factor study is done, then we would pursue OTC.
So generally, yes, you can, but generally we would prefer that tests that are designed for at-home use be submitted as that complete submission. Obviously we have a lot of submissions that we're working through, and from a resource perspective, it is more beneficial both for FDA reviewers and for the public health to have those tests available as quickly as possible. And so it's a much more streamlined process for us to be able to review the complete data set and the complete submission all at once and go straight for that over-the-counter at-home testing authorization, which it is really designed for and will be most beneficial in that setting.
Okay.
Our next question will come from, I believe they recorded their name as Talisha Lee or either Alicia Lee. Your line is open.
Hey, thanks for taking my call. I wanted to get clarification on the antigen template regarding multi-analyte panel. So it's stated that if your device has been previously FDA clear for influenza or other respiratory infections, you should also test at least 10 retrospective positive clinical specimens of each previously clear analyte. So our proposed assay is a POC antigen test, and wanted to double check that this requirement can be met with in-house side-by-side analytical testing.
So you're looking to do the validation for a previously cleared, or for basically for adding SARS-CoV-2 to a previously cleared point of care antigen test that was previously cleared for flu or other respiratory analytes?
Yes, previously cleared for flu. So I'm wondering if that particular template requirement of testing the at least 10 retrospective positive clinical specimens, if that is meant for analytical testing, or is it speaking to a clinical study?
Yeah, that is for analytical.
Okay. And then just to double check, so is there any additional clinical evaluation required for the previously cleared influenza part panel, or are we just able to reference previously cleared 510K data?
So we would want to see those retrospective clinical specimens that we just talked about, are tested in-house, and then you would be able to leverage information from your previous clearance.
Okay, thank you very much.
Yes.
And as a reminder, if you'd like to ask a question, you may press *1 and record your name clearly for question introduction. Our next question will come from Roxanne Chang. Your line is now open.
Thank you. Yes, we would like to add saliva as a sample type to our existing EUA. This is 30 co-validation study. So my question is, for the saliva samples, currently we have a mixture of raw salivas as well as saliva collected using an FDA-approved device. And counting them together, we will have more than 30. And is this something that's acceptable to the agency, or we have to have 30 raw saliva and 30 saliva collected using an approved device?
So I guess the first question would be, when you say an approved device, do you mean one of the saliva collection devices that has an FDA, an EUA authorization?
Yes, that's correct. I'm sorry, yes, EUA.
Okay. So we do, we want to see the test validated the way that you're intending for it to be used. So if you are intending for the saliva specimens to be collected using one of those authorized collection devices, then we would want to see the paired specimens in that device. If your intention is for the test to be used with raw saliva, then we would want to see it validated in that way.
So that means that you would like to see, well, our intention really is to validate raw saliva. So you would like to have 30 paired raw saliva with NP then. Is that correct?
Yep.
Okay. All right. Yeah. Thank you.
We want the 30 paired to be with the specimen as it would be intended to be used.
Understand. Okay. Thank you.
Thank you, Toby. That would be our last question. I just want to thank everyone for participating today. This is Irene here. We appreciate your participation and thoughtful questions during today's town hall. Today's presentation and transcript